Good morning, everyone. So today I would be like, first of all, thanks to the organizers for giving me an opportunity to share my research here. And so I'm directly jumping into my topic today. So I am going to talk about probing facilitated diffusion insights into an optimized target search strategy. Target search strategy is what I mean as mainly in the context of biophysical processes. And this is one of the key steps in the genetic regulation process, all the genetic regulating systems in biophysical systems. So what are those? There is like translation, transcription, genetic editing, and all this. So one can already understand what is the importance of this kind of strategy, or to understand the microscopic picture of this kind of strategy, right? So very schematically, if I have to look at the process completely without looking at the other complexities, just to give a very briefly idea or the physical insights into the process. So I can think of DNA as a polymeric chain, maybe as a rod or a flexible chain, depending on the situations, we will talk about it. And then the protein is there, which actually searches for its target. And finally, it finds the target and goes there and binds it. In chemical physics term, we call it as a reaction diffusion system, where the diffusive mechanism timescale is much, much more slower. Then in that case, it comes back to the diffusive, like we can actually give it by the diffusive framework. So the DNA protein target search, what I mean here, like majorly what I will be discussing about the problems where we can actually describe the process by diffusive framework. Unlike other diffusive processes in biophysical system, DNA target search has a one unique very characteristics. If I just directly put the diffusive framework here, I will find that the rate what I am calculating, that is way more slower than it is actually in the system. So it is almost 10 times faster than other diffusive controlling processes. And how it is actually, what is the mechanism? The mechanism behind this is it is facilitated diffusion, which is actually a combined strategy of 1D and 3D diffusion. It somehow sounds like probably very easy trick, but I can give a very general example of, let's say I have to do a treasure hunt and I am a person who doesn't actually see, like I am blindfolded, but only I can sense it, which in physical terms I would call it as interaction. So I can go through the floor, and then at some point, I can also detach myself from the floor. I actually take a flight and then see where my target is, or sense where my target is. And finally, this is the two-phase system where I keep coming back and forth and then finally find out my target. So that exactly the protein does. So basically, I have two time scale which facilitates each other, and that is why it is called facilitated diffusion, which is one is 1D, which is along the contour of DNA, and another is 3D, which is the bulk diffusion. So that means if I have to actually look at the optimized target search process, that means when there are complexities in the medium and I need to find out what is the time scale and how it is getting optimized or facilitated, I actually need to quantify the rate or the time scale of the process. So this is majorly governed by the interaction coming from the DNA and the protein. And these interactions are usually non-specific because it is coming holistically from the DNA itself. And then at the target, it is very specific interaction, right? So that means this 1D and 3D diffusion, which is actually controlling the target search processes or the target search dynamics, that is driven by DNA protein interactions. So we can always manipulate it and can get the microscopic picture. So in order to now summarize with the mathematical detail, what I can say that the target search time scale can be actually equated to number of search rounds, that is number of times I come back to 1D search. So time scale of tau 1D, which is 1D search time scale, and tau 3D. And then it can be proportional to D3, which means diffusion coefficient at 3 dimension, D1, the other term which is related to coefficient of 1D, okay? So this is very well studied system. There are plenty of research paper experimentally, theoretically, computationally, analytically, and Still now, the overall process of optimized target search, depending on the different system, considering the flexibility, considering the structure of the protein, then considering the medium and all of this, it's kind of the research is ongoing. I have just put some few very basic, like, I mean, it started from this basic theoretical structure, and then we are building on and then getting experimental outputs as well. So with this framework, so major motivation of this to understand the microscopic picture, as I have said, but recently there is recent work by Dudna's group. Dudna actually received Nobel Prize in 2020 for CRISPR-Cas9 kind of protein, which is actually called gene editing tool, which is a complex protein. So I'm not going to actually that work of that, but they have done a genome interrogation study which by fluorescence correlation spectroscopy, and they have found out the dynamics of the CRISPR-Cas protein, which actually goes and binds to the DNA, which is shown here, and in a specific space, okay? So we have considered this problem as a DNA protein target search problem, because here also Cas9 protein has to go there before editing, okay? And that is definitely diffusive search. And from their experimental output, of course, they have also done the theoretical analysis to give the fitting functions and all. And so that is dependent on the Brownian dynamics kind of analysis. And they have plotted, or they have presented their data by plotting the intensity correlation function of different protein complexes, at the same time survival probability. So looking at this, then we wanted to approach this problem with an analytical solution to understand what is going on. And then what we tried to give the protein evolution equation by generalized Langevin equation with fractional Gaussian noise. And then we considered the non-specific interaction which is coming from DNA. We considered that it is, because we are also considering the protein DNA as a rigid rod. So there will be always a cylindrical volume which is very close to DNA, and the protein never gets to go out of this. As a result, so we construct the equivalent Fokker-Planck equation but solve it in the cylindrical coordinate regime, and then we find out the expression for P, probability distribution of protein, and then from there we can easily calculate the survival probability, mean square displacement, survival probability, and those are the analytical expressions, and which shows quite a good agreement with the experimental data what they have observed. Not only that, the intensity correlation function also, with suitably looking at the physical parameters, what we can see that this also kind of reasonably well goes well with the experimental data. The major outcome of this is what we understand, that we have taken two major approximation. Number one, the DNA we have considered as a rigid, because that's the kind of assumption already here. And the second part is we have considered a generalized Langevin equation with a memory term there, considering there is a crowded chromatin medium, which they have mentioned actually. And another important part of this is, even though we call it facilitated diffusion, where time scale is much faster, as I have introduced problem, but at the same time, if you look at this time scale, here it is second, so it is quite slow with respect to usual target search. So we had to, oh my God, okay, so I'll try to quickly go through it. Okay, so what we have found out that in that case, it is, yeah, so where I was, yeah, so majorly that is a chromatin which is heterogeneous time scale, which is a heterogeneous medium. So in order to look at the flexibility and the medium present, so we constructed a simulation and we wanted to do the flexibility effect. So what we do, that this is a simulation setup, and majorly we have constructed the interaction by LJ potential, and we actually look at the snapshots and where we find out the structures look as we find out in the facilitated diffusion case. So then major thing is we found out tau 1D and tau 3D by first passage time analysis technique. So we considered the two structures of DNA where DNA is rigid, another case DNA is flexible, and we considered that the LJ cutoff length scale 2.5 sigma, and inside it is 1D, and when it goes outside it is a 3D diffusion. So in that way, from the simulation trajectory, we can directly track tau 1D and tau 3D and also NS, number of times it comes back to each phase. So once we do that, so then we calculate and plot with the non-specific interaction, changing the epsilon NS in my LJ potential. Once I do it, I get a full picture of tau 1D, 3D, and NS, how it changes. So this is, first plot is tau 1D, and where tau 1D goes increasing when basically tau 1D time scale is increasing with the interaction. When the both the cases, like here down is the target is in the middle, and above target is at the corner at the end. And what happens, the 3D remains almost indifferent, because it is outside the interaction zone. This is as per our model. And at the same time, tau 1D keeps on increasing, which is also very physically intuitive, because once the interaction strength is high, that means that protein is much more closer to my DNA, and so tau 1D sliding will be much more slower. So that will be the dominating stage. And if I plot it K, which is 1 by tau target search, that gives the inverted U kind of shape. But when I consider a flexible chain, considering a similar cutoff, which is an essential part of our model, then what I find out that here, unlike before, here the tau 3D, which is blue, which increases and then saturates, and this is higher time scale than tau 1D. So this is a remarkable effect here. That means the two way the dynamics inside is quite different. What is that? That here it is dominated by tau 3D when I calculate K. On the other hand, for the rigid case, it is dominated by tau 1D. So if I do a comparison plot, when I put the target at the center for a rigid DNA case, I see the plot is the center is K profile is higher. But at the same time, if I compare the rigid DNA versus flexible DNA, where the target is at the center, that it initially increases K for flexible DNA, then it plateaus to its maximum value, which is a remarkable gain with respect to the rigid. So that is why probably DNA is a billion pair, base pair chain, and which is kind of wrapped up, and the protein keeps on searching, but still the process is quite effective, efficient, and precise. So, and also in order to, I will skip this slide because we have looked at that whether the target position change, whether it changes the dynamics, but it does not. So this is the plot for this. So here I would like to show you some video snapshot, which is again another remarkable output from this straightforward, this simulation, where we see that when there's a rigid rod and the green ball is basically our toy model for protein. So what we see that at a particular epsilon ns, what is happening that it is actually doing random walk. At some point it is coming to 1D and then it is going back to 3D. On the other hand, if I look at this flexible chain model, that it was at 3D, then it comes back to the chain, it is getting trapped. That means my 1D search is severely hampered. But at the same time, it is somehow giving a stochastic resonance with the flexibility of my backbone of DNA, and that is facilitating the process in 3D diffusion mode. So this is kind of time series snapshot, and the simulation measure the initial detail of MD simulation setup with the straightforward plot we have taken from this paper, and now we are actually working on to decode that whether any disorder is present in the medium because of the flexibility, and following this kind of the suggestion from a paper which we have found out before, that enhancement of target location rate by stochastic modulation of Brownian motion, which actually talks about the, it is stemming stochastic is resonating with the flexibility or the time scale of the backbone. So then there, in the experiment, there was a D plot, which is, yes, I'm completing here. This is the future goal, that actually in that experiment, they also highlight the diffusion coefficient, which is time dependent, and that has a distribution, which is our further motto to actually look at the process, where we have actually incorporated D time wise in a different distribution, and we actually see the anomalous diffusion kind of behavior coming, and there are some remarkable features also coming out with the with this kind of DNA target search processes. So with this work is my first PhD student Diljit, who has graduated, and now the work is being continued with Atul, and I have one more student in the group, who's Paul, and thank you all for listening. Thank you.
So because of the time lag, we will take one quick question. Anyone have any question?
In several of your slides, there was this thing called nonsense. What is that?
Oh, nonsense is something like, yeah. So it's a protein complex. So it is actually this CRISPR-Cas9. This is actually chemical specificity. So they have a complex which is RNA guided, and that there is a kind of section which goes and binds to the target, like in the protein itself. So they now check the dynamics, changing that chemical specificity where there is no particular interaction present for target. But still it shows the general dynamics, which is diffusive time. So that is why they call it nonsense.
Okay, let's
Okay,
thank
so
ma'am.
please, any question, we will take it later.
You
Thank
can
you so much.
ask her in the lunch session.